A gene is a piece of DNA that provides instructions for making codes for a protein, which gives an organism a trait. Genetic transformation includes the insertion of a gene into an organism in order to change the trait of the organism. Plasmids are small pieces of DNA which help the transformation of a gene from one bacteria to another. The gene GFP is a green fluorescent protein, which is a gene that comes from the bioluminescent jellyfish, Aquaria victoria. pGLO plasmid, which we used, encodes the gene for GFP and the gene for resistance to the antibiotic ampicillin.
After we had streaked all of our plates and labeled both of our microtubes, we started the transformation process the next day. We took them out of the incubator the day after to start the transformation. Our next step was to open the plus pGLO and minus pGLO tubes, and using a sterile transfer pipette, we transferred 250 microliters of transformation solution, calcium, into each tube. Then we placed both the plus pGLO and minus pGLO tubes on ice. Then we used a sterile loop to pick up 2 to 4 large colonies of bacteria from our starter plate that were circular and individual. Then we immersed the loop into the transformation solution at the bottom of the plus pGLO tube. We made sure the entire colony was dispersed in the transformation solution and then placed the tube back into the ice rack. We repeated this entire process for the minus pGLO tube. After examining the pGLO DNA solution with the UV lamp, we took a new sterile loop and put it in the pGLO plasmid DNA stock tube and mixed it into the plus pGLO tube. Then we applied a heat shock by putting both the plus pGLO and minus pGLO tubes into a water bath set at 42 degrees Celsius for exactly 50 seconds. Then we immediately transferred both of the tubes back onto ice for 2 minutes. We then removed both tubes from the ice rack and added 250 microliters of LB nutrient broth to both the minus pGLO and plus pGLO tubes. We incubated both tubes for 10 minutes at room temperature and then gently flicked the closed tubes with our fingers to mix and resuspend the bacteria. Using a new sterile pipette for each tube, we then transferred 100 microliters of the transformation and control suspensions onto the appropriate nutrient agar plates. We then spread the suspensions evenly around the surface of the LB nutrient agar by skating the flat surface of a new sterile loop back and forth across the plate surface. Then we stacked our plates and taped them together with our group name on them and placed them upside down in the 37 degrees Celsius incubator until the next day. The next day, after we took it out of the incubator, we saw little to no growth. However, after the weekend, we saw many colonies grow and glow.
The negative LB had growth, while the negative LB amp had no growth, due to the amp killing the LB. However, when the plasmid was added to the LB amp, there was growth. In the LB amp, ARA, there was no growth and glow. The glow was from the ribosome sugar feeding the bacteria. In conclusion,
In our pGLO lab, the bacteria were transformed, which enabled them to glow. It glowed due to the arabinose sugar turning the operon on. Our -LB and -LB amp did not glow because they did not contain a plasmid or the arabinose sugar. In the +LB/amp plate, the bacteria glowed but did not glow again due to no sugar. In the last plate, the +LB amp and the arabinose sugar plate, it glowed because the arabinose sugar turned the operon on, allowing the green glow gene to be expressed.